parasympathetic division, or craniosacral. Why is it called craniosacral division? It reminds you where the cell bodies of your preganglionics are found, and that would be brainstem nuclei and spinal gray in the sacral regions of the core. And I didn't mention this previously when we talked about lateral horns, but just to throw you off, it does turn out that S2, S3, S4 spinal cord segments do have lateral horns. And in those lateral horns are the cell bodies of preganglionic parasympathetic neurons. What we looked at previously in terms of lateral horns was thoracic and upper lumbar. And there they are very large. The lateral horns are very obvious, not so obvious in the sacral cord segments. But I do have to mention that because we are accounting for both the pre- and post-ganglionic cell bodies. And the preganglionic cell bodies whose axons pass through the pelvic splanchnic nerves, those cell bodies are in lateral horns in the sacral spinal cord segments. So cranio, because cranial nerves carry those preganglionic efferents out of the brain to the targets. Sacral, because we do have sacral cord segments containing preganglionic neuron cell bodies whose axons pass out through pelvic splanchnic nerves. Right. So we have cranial nerves and sacral nerves carrying our parasympathetic output away from the CNS towards the targets. And then sympathetic we call thoracolumbar. It also reminds us where we find the preganglionic cell bodies. Thoracic, spinal cord segments, and upper lumbar. So going back to parasympathetic, top of page 2. Preganglionic cell bodies are in brainstem nuclei in spinal cord segments, and the axons pass through cranial nerves 3, 7, 9, 10, oculomotor, facial, glossopharyngeal, vagus. Oculomotor provides parasympathetic output to what targets? Oculomotor provides parasympathetic output to what? What in the eyes specifically?
Intrinsic muscles of the eye.
Intrinsic eye muscles. Those being the iris, and that changes the diameter of the pupil, and the ciliary bodies, which change the shape of the lens. Is it sympathetic or parasympathetic that constricts the pupils? Parasympathetic. Sympathetic dilates. So during fight or flight, your pupils dilate. Facial provides parasympathetic output to which targets?
Salivary glands.
Submandibular, sublingual salivary glands, lacrimal glands, nasal mucosa. Glossopharyngeal provides parasympathetic output to parotid salivary gland. And I meant to look up otic, but it has something to do with that parotid salivary gland. Vagus provides parasympathetic output to just about everything in the ventral body cavity. All the way down to the bottom of the abdominal cavity. But once you get beyond that into the true pelvis, deep in the pelvic cavity, vagus doesn't quite go that far, so we have to use those pelvic splanchnic nerves to take care of the rest. So 90+% of all parasympathetic output travels through the vagus. And what the vagus doesn't innervate, other cranial nerves do, along with the pelvic splanchnic nerves. Right? So 3, 7, 9 take care of salivary glands, lacrimal gland, nasal mucosa. Pelvic splanchnic nerves take care of a few organs way down in the pelvic cavity. Everything else is taken care of by vagus. Postganglionic cell bodies. Right, so preganglionic cell bodies are in brainstem nuclei, and the axons exit through cranial nerves. And preganglionic cell bodies, again, we're looking at parasympathetic right now, also found in sacral spinal cord segments, S2, S3, S4. Those axons pass through the pelvic splanchnic nerves to reach the postganglionic cell bodies. So now moving on to postganglionic cell bodies. They are found in terminal ganglia and intramural ganglia. Terminal ganglia are very close to the target, but not actually in the wall of the target. And they have names: ciliary, pterygopalatine, submandibular, otic. Notice that the preganglionic, the dark blue, gets close to the target, but not quite there. The light blue, the shaded one, that's the postganglionic. Well, for the vagus, the dark one goes all the way to the wall of the target. And so that postganglionic is very, very short. And there is no ganglion outside of the target organ to name. So these have no names. But collectively, they're all intramural ganglia, which translates to within the walls. Intramural ganglia. So what are the two types of ganglia that contain your postganglionic cell bodies? Parasympathetic division? Terminal and intramural. And which one is actually in the wall of the target? Intramural. Next is sympathetic. Preganglionic cell bodies are in the lateral horns of the thoracolumbar spinal cord. Axons of these preganglionics pass through white rami of rami communicantes before entering the sympathetic trunk. This is part of the sympathetic trunk. So we only see 1, 2, 3 ganglia. There's 23 altogether. And they start up in the cervical region, and you have a left and a right, a trunk on each side, and it goes all the way down to the pelvic region. So it's a really long chain of ganglia, all part of the sympathetic division. And so it has many different names: sympathetic trunk, sympathetic chain, prevertebral or paravertebral ganglia. Sympathetic chain ganglia, sympathetic trunk ganglia, pre or paravertebral ganglia. And here's the whole thing. Here's your 3 cervical sympathetic ganglia, and they actually get named superior, middle, inferior. And then thoracic, lumbar, sacral, and 1 coccygeal at the very bottom. There are 3 paths that the preganglionic axons can take, and these are the 3 options. Option 1, the preganglionic axon goes through the ventral root, spinal nerve, white ramus, and into the sympathetic trunk ganglion at the same level as the cell body of that preganglionic axon, or preganglionic neuron. So at the same level, meaning once the preganglionic gets into the trunk, it doesn't go up or down, it synapses immediately at the same level. Option 2, same thing, out of the cord, ventral root, spinal nerve, white ramus, into the trunk, but rather than synapsing at the same level, you can ascend. You can ascend 1, 2, 10, depending on how many are above ganglia, or descend. Right, so many options, but it's just not at the same level. Somewhere above or somewhere below. In this example, we're just going up 1. Third option, axon leaves the cord through the ventral root, through the spinal nerve, through the white ramus, and not stopping in the trunk, but going through the sympathetic trunk to finally synapse on a postganglionic cell body in a sympathetic ganglion, somewhere outside of the chain. So with options 1 and 2, the preganglionic synapses on the postganglionic within the chain, either at the same level or higher or lower. Option 3, you don't synapse in the sympathetic chain ganglia, you go through the chain and synapse outside of it in a sympathetic ganglion. If you're taking option 3, then you're passing through splanchnic nerves to reach the ganglia. And the type of ganglia is called collateral. So this is a collateral ganglion right here, shown in option 3. And they have names. One example is the celiac, and that's the name they're giving you as just one example of many. So here's another look at what we just saw. Preganglionic leaves the cord, goes through the ventral root, goes through the white ramus, goes into, but through, without synapsing, through the chain, continues as splanchnic nerves before finally synapsing in a collateral ganglion, such as celiac or superior mesenteric. And then down from lower thoracic, upper lumbar region, you have the lumbar splanchnic nerves, which are another example of option 3. They carry preganglionic axons that have gone through the chain without synapsing, continue through these splanchnic nerves, and into the inferior mesenteric ganglion. Okay, so celiac, superior mesenteric, inferior mesenteric, those ganglia are examples of collateral ganglia. They are outside of the sympathetic chain. Sacral splanchnic nerves carry postganglionic fibers, not preganglionic, similar to the sympathetic nerves extending from the cervical ganglia. So when you look down at the bottom, notice that this green axon is shaded. It's light in color. That means it's a postganglionic, just like all of these. These are all post. The dark green coming from the cord, that's pre. Right? So you can see examples of preganglionic synapsing on postganglionic in the trunk or chain. Right? So here's superior cervical, middle, inferior. And these ones in the thoracic region don't have names, but here are many examples of pre-synapsing on post in the sympathetic chain. And then from the chain, the postganglionic takes you all the way to the target. Right? So we said earlier that preganglionic sympathetic tend to be short. They're just going from the cord to the sympathetic chain, or many of them are. And then from the chain, you still have a long way to go to the target, making the postganglionic really long. Over here, it's not quite as distinct as up here, where you have a really short pre and a longer post. These are sort of medium in length, both pre and post. So notice the dark green goes past the chain, all the way to the collateral ganglion. And then the postganglionic takes you from the ganglion to the target. So going back to where we left off, the sacral splanchnic nerves. These nerves are not like the greater and lesser splanchnic, or even the lumbar splanchnic. The difference is greater, lesser lumbar splanchnic nerves carry preganglionic fibers. Sacral splanchnic nerves carry postganglionic fibers. That's the difference. Next is postganglionic cell bodies. So where do you find them? Because now I'm just reiterating what I've already covered. Where do you find postganglionic cell bodies? Either in the sympathetic chain. Here, 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 here. Those are examples. Here, more examples. Or collateral ganglia. Cell bodies of postganglionic sympathetic neurons are either in the sympathetic trunk or chain, or you go through it and past it onto collateral ganglia. The postganglionics extending from the superior cervical ganglion primarily innervate structures in the head. From the middle and inferior cervical ganglia, those postganglionics primarily serve organs in the thoracic cavity, heart and lung. And notice the name given to these sympathetic nerves is the cardiac and pulmonary plexuses, right? Because they sort of intermix, just like a sacral plexus or a lumbar plexus. Collateral ganglia. These postganglionic fibers primarily serve abdominal pelvic organs. So from the celiac ganglia, we innervate gallbladder, liver, stomach, spleen, adrenal gland. From superior mesenteric, mesentery in general refers to the intestine. So from there we innervate large and small intestine. From the inferior mesenteric ganglia, large intestine and rectum. So that covers pretty much everything in the abdominal cavity, but it doesn't cover some of the organs way down in the pelvic cavity. And so again, to reach those organs, we use the sacral splanchnic nerves. So those sacral splanchnic nerves and the pelvic splanchnic nerves of the parasympathetic division are innervating similar structures way down in the pelvic cavity. Third option. Okay, so again, we're looking at where we find postganglionic sympathetic cell bodies. We find them in the sympathetic trunk. We find them in the collateral ganglia. And third, the adrenal medullae, which is plural for adrenal medulla. So notice we have one very long preganglionic. It goes all the way to the inside of the adrenal gland. In here. Preganglionic takes you all the way to the target cells. So this is unusual. So we are in the middle of the adrenal gland. This outer layer is the adrenal cortex. This inner layer is the adrenal medulla. And what we're innervating, and I'll make this a little smaller, are the equivalent of postganglionic sympathetic neurons. Which makes sense because these are pre, right? That dark green one that goes all the way from the cord to the adrenal gland. Oops, right here. That's one long preganglionic. These modified postganglionic neurons are called chromaffin cells. They look like endocrine cells, they function like endocrine cells, they secrete hormones like endocrine cells, but they're actually modified neurons. Now, if this was a typical postganglionic neuron, what would it secrete as its neurotransmitter? Right, so remember, for preganglionic, sympathetic or parasympathetic, they secrete acetylcholine. The postganglionic, for parasympathetic, also secretes acetylcholine. The difference is ACH binds to nicotinic receptors at the ganglion. ACH binds to muscarinic receptors at the target cell. Now for sympathetic, the pre does release ACH, but the post releases norepi. So what do you suppose these chromaffin cells secrete? Norepi. And within the adrenal medulla, lots of blood vessels. So the chemicals being secreted by exocytosis from these chromaffin cells, rather than being used as a neurotransmitter, goes into the blood vessels and acts as a neurohormone. And it turns out 80% of the chemical signals secreted from the chromaffin cells is epinephrine. And about 20% is norepinephrine. But they bind to the same receptors. They more or less give you the same effects. The only difference is the binding affinity. Epi has a stronger binding affinity for beta adrenergic receptors. Norepi has stronger affinity for alpha adrenergic receptors. But big picture, they both bind to both types of receptors. So we can sort of look at them as a unit. In fact, I did give them a combined group name, which is what? Catecholamines. And as a group, going back to chapter 11, we categorized norepi and epi and dopamine as biogenic amines. Yes, they are catecholamines, but they fall under the umbrella heading of biogenic amines. On the board, they're pretty much showing you right here. Right, so the pre-synapses on the post at autonomic ganglia, either the sympathetic chain ganglia or collateral ganglia. And then the third possibility, where you would find a postganglionic sympathetic neuron, in the adrenal medulla. But these are very modified postganglionic sympathetic neurons, again called chromaffin cells. They secrete epi and norepi. What's stimulating these cells to secrete epi and norepi? The release of ACH from the preganglionic sympathetic fibers. Right? So it's just like this. Pre secretes ACH, which stimulates the postganglionic to release its neurotransmitter, norepi. But here, things are modified a bit. It's not a typical postganglionic neuron. It's a chromaffin cell. But the preganglionic still releases ACH, and that stimulates the chromaffin cells to release epi and norepi. Next is part 3, ANS physiology. Visceral reflexes are similar to somatic reflexes, but motor output is a 2-neuron chain. Visceral reflexes are involved in visceral reflexes involved in digestive functions take place outside of the spinal cord, in the enteric nervous system. I don't even think I mentioned the ANS this semester. There is what's called the little brain of the gut, a complete network of neurons, including sensory neurons, interneurons, and motor neurons, completely contained within the walls of the gut, stomach, and intestines. It's called the enteric nervous system. It's responsible for the visceral reflexes that regulate digestive activities in those organs, including the stomach and intestines. So we'll cover that specifically in 431. Otherwise, how is the visceral reflex different than somatic? So the one thing that we already mentioned, in a somatic reflex, the efferent pathway is a single efferent neuron. The motor neuron takes you from the CNS to the skeletal muscle. Right? And if the target is skeletal muscle, it must be a somatic reflex. But if the target is anything other than skeletal muscle, then it must be a visceral reflex. And then anatomically, the difference is it's a 2-neuron chain to get from the CNS to the target. Right? You got your pre and your postganglionic neurons. Another difference, typically for a visceral reflex, not only is the effector organ something other than skeletal muscle, so a visceral effector, the sensory information coming in is typically from the viscera as well. So what type of a receptor is responsible for providing input? Primarily interoceptors. Now, it is possible for a somatic sensory input to result in a visceral reflex. It's possible for visceral sensory input to result in a somatic reflex. And that's why I use the term usually or mostly, but not always. Usually for a somatic reflex. The target, of course, is muscle. Right? If it's muscle, that makes it a somatic reflex. But the input could be something other than somatic sensory in nature. And likewise for a visceral reflex. Definitely the target is visceral in nature, something other than skeletal muscle. But the stimulus doesn't have to be visceral, doesn't have to be from interoceptors. It could be somatic. Neurotransmitters and receptors. All autonomic preganglionic fibers are cholinergic fibers, which means they secrete what? Acetylcholine. And at the autonomic ganglia, that ACH is binding to nicotinic receptors. All parasympathetic postganglionic fibers are cholinergic. They all release ACH, just like the pre. Most sympathetic postganglionic fibers are adrenergic. Most secrete norepinephrine, but not all. What makes it really confusing, unfortunately, is that the other neurotransmitter that postganglionic sympathetic neurons sometimes use is ACH. So we do have cholinergic postganglionic sympathetic fibers. They are not all adrenergic. Most are, but not all. For example, the postganglionic sympathetics that innervate sweat glands. They are cholinergic. They release ACH. But just because it uses ACH as a postganglionic doesn't mean it's parasympathetic. That's not what determines whether it's sympathetic or parasympathetic. In addition, we also have some sympathetic fibers that release nitric oxide. So we have adrenergic sympathetic fibers, which is most of them, cholinergic sympathetic fibers, some of them, and nitroxidergic sympathetic fibers, just a few of those as well. Primarily blood vessels are using those nitric oxide receptors. As far as cholinergic receptors, you've got nicotinic and muscarinic, which we've looked at. And then for adrenergic receptors, alpha and beta. And we'll look more at these next class. Neurotransmitter effects at the target cells. So on the next few slides, 14, 15, I'm giving you some, not all, but some specific effects. Right, so before we get there, in general, for the parasympathetic division, it carries out maintenance functions, stores energy, conserves energy. We consider this to be rest and digest. So the anatomical nickname for the parasympathetic division is craniosacral. Reminds you where you find the preganglionic cell bodies. The functional nickname for parasympathetic division, rest and digest, reminds you of what it does, right? Big picture. So you're doing one of two things. You're either in rest and digest mode, or you're in fight or flight mode, one or the other. So where do we spend most of our time on a daily basis? In rest and digest mode, where parasympathetic output dominates. Sympathetic is turned down, parasympathetic is turned up. But whenever there's a threat, crisis, right, something that initiates the fight or flight response, the stress response, then the dial gets turned down on parasympathetic, and sympathetic dial gets turned way up. And then we go from maintenance and storing energy to mobilizing our energy reserves and using lots of fuel, specifically for skeletal muscles. Fight or flight is all about preparing the body for physical action, to literally fight or run. Skeletal muscles are heavily involved in this response. Their metabolic rate goes way up. They need a lot more blood flow, a lot more nutrients. Sympathetic nervous system is going to mobilize those nutrients. It's going to tell the liver to release glucose, tell adipose to release fatty acids. So going back to parasympathetic for a moment, these are general effects. Here's some specific effects. At the heart, it keeps heart rate low, thereby keeping blood pressure low. Constricts the iris. Responsible for emptying the bladder. Responsible for digestive functions. Motility refers to muscular contractions within the gut, the stomach, the intestines, and then secretion as well. Right? Lots of glands that are secreting, lots of smooth muscle in the walls of the gut contracting. Parasympathetic is excitatory to the urinary system and the digestive system. Sympathetic, on the other hand, is inhibitory for urinary and digestive functions. So we divert blood flow from those areas to skeletal muscle and cardiac muscle during fight or flight. So again, big picture. Sympathetic general effects prepare the body to cope with emergency, a threat. This is a time of increased muscular activity, running or fighting, fight or flight. A few specific effects of sympathetic output. Increases heart rate, as norepi stimulates beta-1 receptors on cardiomyocytes. Signals adipocytes to break down their triglycerides and release fatty acids into the blood that the muscle can take up and use by using beta-3 receptors on the adipocytes. Blood vessels have alpha-1 receptors, including blood vessels in the skin. Alpha-1 results in constriction. When epi and norepi stimulate alpha-1 receptors in blood vessels, you get constriction. I think my slide numbers are off. I wonder if I'm using the wrong slides. 15, 16. It's like one off. Maybe the one on, I'm sure the one on D2L is correct. This one might be off. So for mine, 15 is autonomic tone, not 16. But anyway, autonomic nervous system is always active. Endocrine system is always active. Remember, they don't function like a switch. It's not on or off. They're always on. It's just a matter of how much are they turned up or down. Think of it as a dial. So your sympathetic fibers, always releasing norepi. Parasympathetic fibers, always releasing ACH. How much is the question? So right now, during rest and digest, all of your target organs are primarily receiving sympathetic stimulation. Right? Very little norepi coming from your sympathetic fibers. Lots of ACH being released from your parasympathetic fibers. This includes parasympathetic output through the vagus nerve to the heart. So when you have constant stimulation, we call that tone. You have parasympathetic tone to the heart, as an example, keeping your heart rate really low. We know this. There's a very simple experiment we can do to prove this. Which nerve is responsible for all parasympathetic output to the heart? Vagus. So you cut the vagus nerve, and guess what happens to heart rate? Shoots up. Goes up to 100 beats per minute. So your resting heart rate could be 65. Cut the vagus nerve, eliminate that parasympathetic tone, jumps up to 100. Now if you need to get your heart rate up above 100, what are you going to have to do besides turning down the dial on parasympathetic? Turn up the dial on sympathetic. And that's pretty much how the system works. If one's getting turned down, the other one's getting turned up, and vice versa, right? Because they have opposing effects. There was only one exception to that rule, and that's reproductive organs during the sexual response. There they actually work together. But otherwise, one is excitatory, the other is inhibitory. We have examples of sympathetic tone as well. Sympathetic stimulation of blood vessels keeps them partially constricted all the time. We don't have sympathetic and parasympathetic innervating blood vessels. We can't use one to constrict, one to dilate. We have that in the pupils of the eye. We don't have that with blood vessels. Because remember, blood vessels was an example of a target organ that does not receive dual innervation, sympathetic only. What were the other examples? Adrenal gland, sympathetic only. Blood vessel, sympathetic only. Sweat gland, sympathetic only. Erector pili muscle, sympathetic only. So if you're only using sympathetic to control blood vessels, you have to start in the middle. You can't start fully dilated nor fully constricted. You start partially constricted. And then from there, if you turn the dial up, constriction. You turn the dial down, dilation. But you have to start in the middle with sympathetic tone. We'll stop there.